Hi, everyone. Thanks so much for coming to today's seminar featuring Dr. Kyle Lowe. He will be discussing and presenting his work related to creating human blood vessels to study vascular development and disease. I think it'll be a really compelling topic. Please keep your audio muted, and we will be recording this session just so you are aware. But first, we have a few advertisements of some things that are going on. Just first want to thank our education committee who help us put on these seminars. We're really blessed through their monthly and regular support of our activities. If you are interested in participating in this committee, please let us know. We're always looking for new members. Our next topic, we're going to take some time over the summer as a little break. We know everybody has a lot going on, and we'll pick things back up in September on the 14th, featuring a talk from Jason Cunningham related to using systems thinking to support community collective action in healthcare. Really interesting topic. You can scan that QR code there to sign up for the next seminar. Registration is now available. And then another topic that we have scheduled is in December. We will have some other sessions between then and now, but we are still confirming them. But a really interesting topic from Dr. Stanley Key, who has some wonderful areas of interest related to gene editing and developmental disorders. That registration is now available, but a really cutting edge topic for those that are really interested in this space. We want to make everyone aware of the Summer Scamper. MCHRI has a team. We would love for you to consider supporting our team, whether it's running with us or walking with us or supporting our team financially. Whatever you so choose, we would be blessed by your participation. That's coming up on June 21st, right around the corner. And just for everybody's knowledge, that these funds that are raised through the Summer Scamper go directly to support MCHRI's activities. So if you have received support from us or resources, or just want to support our efforts writ large, please consider doing so. And then please mark this on your calendars. This is a great opportunity for our research community to come together. It's a Stanford Medicine Children's Health community event, all focused on research. Those from the hospital, as well as those from our School of Medicine community, can get together for over lunch and learn about different aspects that are going on, learn about case studies where people are working together to kind of promote and advance uh, clinical research and how our hospital supports those endeavors. So please consider attending. We'd love for you to support it. Please scan that QR code to register so we know that you'll be coming. And that'll be hosted at the Center for Academic Medicine's Grand Round Room. And we hope to see you there. Another really big activity coming up, as we've hosted annually in October, our annual research symposium. This year will be focused on correlations, causation, and cures. We'll have a number of sessions. Registration is now available, and we have a keynote speaker from the University of Alabama at Birmingham, um, in Dr. Alan Tita. He is an endowed chair of OBGYN as well as the Senior Associate Dean of Global and Women's Health there. So we're really excited by his talk. But now want to turn our attention as to why we're all here today. Um, and I will pass this off to Matteo, one of our education committee members, to introduce Dr. Lowe. Matteo, take it
Right.
away.
Thank you, Grant, and I'm super excited and really an honor for me to introduce our next speaker of this seminar series, Dr. Kylo. Just to give a brief introduction, Kyle is an associate professor of developmental biology and also the Anthony Di Genova Endowed Faculty Scholar. here at Stanford University. Kylie is really globally recognized as a massive pioneer in stem cell biology and recently received a fantastic award, the 2026 International Society for Stem Cell Research Early Career Impact Award. That's massive. Congratulations. And his groundbreaking research really focused on developmental roadmap of the human body, mapping embryonic stem cells, how embryonic stem cells branch into many different cell types of the body. And most importantly, Kyle has uncovered with a precise and beautiful science the signaling pathways required to guide these stem cells into nearly pure homogeneous population of functional human tissues. So really, the law laboratory is bridging the gap between fundamental developmental biology and regenerative medicine. So such a great honors, and I'd like to please join everyone to give a warm welcome to Dr. Kylo.
Thank you, Matteo. That's very flattering, and I'm also very happy to see lots of friends and colleagues in the audience, such as Minx, Matteo, Jess, also people whose work I've followed a long time, like Li Wang, Maya, and Jeremy Height, actually. So this is very informal today, so please feel free to interrupt me with any questions along the way. Our lab, as Matteo mentioned, has mapped how human pluripotent stem cells differentiate into over 20 different cell types. At each step of differentiation, we ask, what are the intermediate progenitors that stem cells have to go through to become a certain cell type? And what are the combinations and timings of signals that have to be turned on or off to affect each transition? You might think this sounds really boring because much of this is already known, because I'm just telling you about gastrulation and early development, but I hope to convince you today that there's maybe one or two things left to figure out about gastrulation. So we've come across the two general principles. First, at each step of differentiation, progenitors face a fork, become one cell type or another. We found that by activating a signal to tell the cells go one way, of equal importance, blocking the signal to prevent it from going the other way, we could really force stem cells to become a very pure population of a given cell type, as Matteo alluded to. Another principle we found is that a single signal like Wnt could specify 3 totally different cell types over 3 days of differentiation. In fact, I think it was Minx who told me in grad school that timing is everything, or something along the lines of that aphorism. So as I'll show you in work today, if you're a stem cell and you encounter Wnt, it tells you to become a certain cell type, the primitive streak. But one day later, if you see the same signal, it tells you to become something totally different. I think that's really important because many of us who work in labs know that there's many papers saying that a certain signal induces one cell type or inhibits another. And obviously both are true. It's timing, that's everything. By combining these principles, we can guide stem cells to become very specific kinds of cells. So today, I'll talk about 2 different things. First, our efforts to create human blood vessels, including arteries and veins from stem cells, and their various uses to study development, and also BSL-4 viruses. Maybe have more interest to Jess and her world. The second is about the brain, and I'll first begin on blood vessels. So just to orient you in this roadmap, blood vessels come from the red group of cells, which are the mesoderm. So this project had 2 parts. First, we figured out how to turn stem cells into the building blocks of blood vessels, artery and veins. We discovered something really weird. So it's well known that certain viruses I'll tell you about infect human blood vessels, but we found that specific viruses, specifically Nipah virus, did not affect all blood vessels equally, but actually preferentially attacked arteries and had a really weird effect on arteries. And to our knowledge, this is the first time it was discovered that there's a certain kind of virus that preferentially affects a certain kind of blood vessel. So let me take a bit of a step back and tell you about something quite different from Matteo's intro on development stem cells, which are BSL-4 viruses. So these are among the deadliest viruses on Earth. This is not a hyperbole. These include Ebola virus, of which there's actually a raging outbreak right now, sadly, in the Democratic Republic of Congo and Uganda, and Nipah virus, which I'll focus on more today. So Nipah virus has a 57 to 59% fatality rate. If you think about that, that's absolutely staggering, right? 3 out of 5 people die. And for context, this is over 100 times deadlier than COVID-19, which is caused by BSL-3 coronavirus. So what does it look like to work in the BSL-4 lab? Well, it looks very different than I imagine my lab or many of yours. You have to wear a full body, a positive pressure spacesuit. You get air delivered from this hanging air line from the ceiling, and you basically have to plug in whenever you want to breathe and unplug when you want to walk around. And I won't spend much time on this today to get more to the biology part of it. So what are the effects of these viruses on blood vessels? So we know that these viruses like Ebola and so on, they cause vascular diseases, right? Think of Ebola, you think of hemorrhage and other vascular dysfunction. But you know that there's multiple kinds of blood vessels, like arteries and veins. So we thought, can we first make human artery and vein cells in a Petri dish and then use them to study what these viruses do? So our hypothesis was that in development, there must be a branch point where a common mesoderm progenitor can become an artery or a vein. And we thought, if we just can discover the signals that tell a cell become an artery versus a vein, that by activating one signal and blocking the other, we could really force a progenitor cell to only become either an artery or a vein very specifically. And that's exactly what we did. So in this work and unpublished developments of it, we basically mapped out how to start from a human pluripotent stem cell in gray at the top and how it becomes an artery or a vein. We can now make over 90% pure batches of human artery or vein cells. This occurs in just 3 to 4 days. So I hope you appreciate this is very, very fast and very, very efficient. This occurs in fully defined media, so no need for serum. It occurs in monolayers, so no need for 3D aggregates. And it's completely affected by just extracellular signals that we add to the media by activating or blocking different developmental pathways. And to make these cells, we took a developmental biology approach of figuring out at every step along the way, what are the intermediate progenitors the stem cell has to go through and the signals have to turn on and off. And this work was done closely with my colleague Lei Tang Ang, who's now an assistant professor of urology at Stanford. So now let me walk you through the various steps and how our lab develops these differentiation protocols. So the first step in development is when a pluripotent stem cell becomes one of two cells, the ectoderm in blue that makes the brain or the skin, and the primitive streak in yellow. You may not have heard of the primitive streak before, but this is what gives rise to endoderm and mesoderm. So 4 signals induce the primitive streak: TGF-beta, BMP, FGF, and Wnt. Of equal importance, all 4 of these signals actually block the formation of the ectoderm. And that's what I was alluding to earlier, that at these branch points, there's signals that induce one cell type, these that block another. So this is all well known in an embryo in vivo. So we thought, what happens if we take stem cells in a petri dish and add these 4 signals in vitro? This actually succeeds in generating an over 98% pure population of these yellow cells, the primitive streak. As you can see here, MCHRI is a gene that identifies the primitive streak, and over 98% of the cells express a primitive streak marker. So whenever you see data like this, you should be very skeptical because there's so many claims in the stem cell field of people making very pure batches of cells. So we want to use multiple independent methods to confirm this result. So we turn to single cell RNA sequencing. And as you can see, every single dot represents a single cell, and we can create an almost completely pure population of these primitive streak cells expressing MCHRI here in purple. And I want to add that actually over 30 labs around the world have actually reproduced this result. And if you or your lab are interested in trying this out, we'll be more than happy to help you. So having made really pure batches of these early primitive streak cells, we then went on to ask, how do they go on to make the 2 major kinds of mesoderm? There's a practical mesoderm in purple that makes the somites. So these are, for instance, the bone, cartilage, and skeletal muscle. And there's a lateral mesoderm in orange. So this makes the blood vessel. So just focus on the lateral mesoderm in orange for now. We found, again, this is a simple lineage split controlled by 2 competing signals. Wnt in purple makes the practical mesoderm. BMP in orange makes the lateral mesoderm, and each class inhibits the other. So this is how cells make a really sharp decision. We thought, if this is true, how can we make the orange cells? We thought maybe we should add BMP, block Wnt. So Wnt I means inhibit. And of course, it's not that simple. We have to inhibit a few other signals. But we can now create almost pure batches of lateral mesoderm cells in just 2 days in a petri dish, expressing this lateral mesoderm marker, KDR. So I hope that this illustrates to you that by understanding these quite simple lineage branch points, we can really command and control stem cell differentiation very precisely. So having made these lateral mesoderm cells, we then asked, how do these go on to make artery versus veins? And again, we found that this is a lineage split controlled by 2 competing signals. TGF-beta in red makes arteries, PI3K in purple makes the veins. So to track differentiation, we need marker genes. So we identify arteries by expression of a gene called SOC17. Familiar to many of you as maybe an endoderm marker, but it's also an artery marker. And with the lab of Matt Porteous in pediatrics, we developed a SOC17 2A and plum stem cell line. So when cells become arteries, they'll glow red. Conversely, the NR2F2 gene is a vein marker, so we make an NR2F2 GFP reporter, so when cells become veins, they become green. So how does this work? First, we made arteries. So starting from these mesoderm progenitors, if we activate the artery signal TGF-beta, and of equal importance, block the vein signal PI3K, we can now make an over 95% pure artery cells. To walk you through, these cells express the endothelial marker V-cadherin on the y-axis and the artery-specific marker SOC17 on the x-axis. So these aren't just endothelial cells, they're artery-specific endothelial cells. And we can make these cells with almost complete purity. I think that's because when we add the artery signal, we block the vein-inducing signal PI3K, which is really, really important to force the stem cells to go down one way. Reciprocally, we can now make veins by activating the vein signal PI3K and inhibiting the artery signal TGF-beta. We can now make over 85% pure vein cells expressing the pan-endothelial marker V-cadherin, but now the vein-specific marker NR2F2. So what do these cells look like under the microscope? We see these artery cells from these beautiful matted networks expressing V-cadherin in the cell surface. But in the nucleus, they express this intense nuclear SOC17. Conversely, the veins also express this general endothelial marker V-cadherin, but now in the nucleus, they express NR2F2. I hope these images reinforce how pure these cells are. So if you look at the DAPI, which are all the nuclei, you can see that virtually almost every single nucleus expresses SOC17. The vein differentiation purity is slightly lower, and you can see occasional rare cells that are present here, but are negative for NR2F2. But I hope this really will convince you that these are really almost pure batches of artery and veins that we can make, as shown by flow cytometry and now by immunostaining, and also by single-cell RNA sequencing. So if we take these artery cells and we don't purify them at all, we look at the whole population, over 90% of the cells express V-cadherin, this pan-endothelial marker. They also express the artery-specific marker SOC17, but they express almost none of the venous marker NR2F2. These are really artery-specific endothelial cells, and the way we make them is by repressing the venous marker signal PI3K. Reciprocally, we can now make veins, which 90% express the V-cadherin. They express low to no levels of SOC17, but they express high levels of the venous marker NR2F2. So you might ask, if they're 90% pure, what the remaining cells, which I've outlined in brown, we're not quite sure what these cells are, but these are, we believe, mesenchymal cells. We're still trying to figure them out. I also just want to add, if you find this data interesting, in our lab website, you can find an online portal where you can type in the name of your favorite gene and its expression will show up. So for instance, whenever Matteo gives a talk and he says a gene that I think is interesting, I'll quickly type it into this portal and see if it's expressed in arteries versus veins.
Kyle, may I ask a question?
Yeah, please. It's
What
very informal.
about
Anyone should feel free to unmute and ask questions.
ERG as a marker? Does it distinguish between artery versus veins, or is it considered more a pan endothelial marker? Like here, if you put ERG, would you also have a
yeah.
Mhm.
Yeah, so which genes are you interested in? So ERG is a pan-endothelial transcription factor, and it's like many people use it as like the single best marker of endothelial cells in vivo because it's like a nice nuclear, unlike
But would you
V-cadherin.
not see any difference between the artery or the vein generated?
Yeah, that's a good question. They are both positive. The level of ERG is slightly different in artery versus veins, but it's still extremely high in both, and we use it to identify both.
Okay, thank you.
Yeah, and if you have any questions, you can look at the website. If anyone has any questions, I'm also happy to take them now. Yeah, feel free to unmute or put them in the chat. Happy to walk through things. So this is meant to be maximally useful to everyone here and happy to collaborate or share resources with anyone. So a big question to stem cell field is reproducibility, because often you read a paper, it seems great, but you try it in your own lab and things doesn't work at all. And I'm very pleased to say that 12 different labs around the world have now reproduced this artery versus vein method. 5 of us have come together in this paper, the journal Star Protocols. And you can see that we can make over 90% pure arteries and over 70% pure veins. And we're happy to help your lab set it up too. I see there is one question from Tao. What cell types are they? Can you specify a bit more what cell types, or what do you mean by cell types? Or maybe I can continue and Matteo, what do
Or
you think?
maybe I
Please
could.
use for.
Oh, hi,
Oh,
Tao.
I think like small muscle cell types, fibroblasts, endothelial cells, what about the cell types?
Yes, these are specifically we're making endothelial cells that express the pan-endothelial marker VE cadherin. So we have not yet made fibroblasts or smooth muscle cells, but I know that our colleagues are trying to generate them as well, and that's a really important goal for the field to make the supporting cells for blood vessels. Jessica asked a question about the reporter cell lines. Yep, these are monoclonal cell lines, so they're single cell clones taken from a whole edited population, and anyone we're happy to distribute these lines to. We've confirmed the mycoplasma negative. Many labs around the world have received them and have used them for different studies. In fact, Matteo, I think someone actually asked me the NR2F2 line for actually synthetic embryo work because NR2F2 is in some kind of trophectoderm population. And SOX17 obviously is the hypoblast marker. So always happy to share these lines. We're not secretive. We want science to go quick. We just want science to go quickly. About reproducibility, I should also add that our lab has now published a 30-page step-by-step protocol in the journal STAR Protocols, how to make these artery and vein cells. This goes to the level of which catalog number you should order, like for growth factor and small molecules, when you get them, what buffer do we suspend them at, what concentration, how much to add. And the reason I'm actually the first author of this paper is there's actually so much work to compile this. I ended up writing this. So if there's any errors in this protocol, you can clearly attribute them to me. So you can find this online. It's an open access protocol, and I'm happy to answer questions. In fact, someone from Switzerland emailed me yesterday with some questions about it, which I responded to. So another key question is whether these cells are functional. In stem cell research, you often make cells with the right markers, but then they might not function the way you want them to. So the function of endothelial cells, of course, is to build blood vessels. So you took the stem cell-derived artery and vein endothelial cells and mixed them in a 3D hydrogel. They form these beautiful 3D vascular networks. And we're now working with tissue engineers at Stanford, like Mark Schuyler Scott, to use them to try to vascularize synthetic tissues. I know Matteo has been interested in vascularizing his endometrial layers, and we were very happy to interact and collaborate with people. These also build blood vessel networks in vivo. So my colleague Lei Tang, when she injected them subcutaneously in mice, both the stem cell-derived artery and veins both formed blood vessels. So it's at the end of the endothelial part before I move to disease. So if there are any remaining questions about differentiation, I'm happy to take them. If not, maybe I'll move on. So we use these blood vessel cells to study BSL-4 viruses. So today I'll focus on Nipah virus, which some of you may not have heard about. But Nipah virus has a 57 to 59% fatality rate, which is really horrifying if you think about it. There are no FDA-approved treatments or vaccines for this virus, and it is actually one of only 2 BSL-4 viruses that's known epidemiologically to spread airborne between humans, which is really quite horrifying and very different from Ebola, for instance, which only spread by close contact. You might ask, where is this virus? So sadly, there are almost annual outbreaks in India and Bangladesh, more specifically often in the Kerala province of India. I hope I'm pronouncing that correctly. So in the newspaper, if you read of an ultra rare deadly virus outbreak in India or Bangladesh, it's often Nipah virus. Now, though you may not have heard of Nipah, one thing to know is that it enters cells through the receptor protein at the cell surface called FNB2. I just want you to keep that in the back of your mind because that'll become quite important a little bit. Now, though you may not have heard of Nipah virus, if you watched the movie Contagion in 2011 with Kate Winslet and Matt Damon, this is actually the virus from Contagion. So you might think this is a bit of a tall tale, but on Amazon Prime, I have actually gone through every single second of Contagion. And you can see at this exact moment, they have a computer in the CDC where they're showing the genome of the fictional virus in the movie and the genome of Nipah virus. And this DNA alignment software is green, indicating that they're homologous. You might ask, what's this number next to Nipah virus? If you put it on Google, this is the actual NCBI accession number for Nipah virus Malaysia variant. First, when I know there's a lot of angst among grad students now, like what jobs they're going to get. And I was telling them in Hollywood, they obviously have large numbers of PhDs that are fact checking all of these movies. So I think that there will be jobs for a lot of people. Of course, that's somewhat whimsical. So the real reason that we worked on Nipah virus was not because of the movie Contagion, but because of its entry receptor called FNB2. So I'm showing you here the levels of FNB2 in cells we're making from stem cells. And you can see that FNB2 in red is expressed at 10 times higher levels in arteries as opposed to veins. In fact, this is actually very well known in the vascular field. And actually the first paper ever in 1998 by David Anderson that found that arteries and veins are molecularly different actually found FNB2 already is higher in arteries versus veins. So it's well known in vivo, and we're just reproducing it in vitro. We thought that's really cool because people once thought that these viruses would infect all blood vessels equally. We thought maybe through the lens of Nipah virus, it would preferentially attack arteries because arteries are full of its receptor on the surface, whereas it might just ignore a vein because it doesn't really have high levels of its receptor. So indeed, that was the case. So working with Joe Prescott's lab, the BSL-4, he found the authentic Nipah virus replicated to over 10 times higher levels in red in arteries as opposed to veins. And I hope that you'll appreciate the stark order of magnitude difference because to our knowledge, this is the first demonstration of an artery bias virus, and that actually raises really interesting implications for using Nipah virus for vector gene therapies if you want to preferentially target genes to arteries as opposed to veins. So you might ask, well, Nipah replicates higher amounts in arteries, but what's it really doing? And again, we found something really weird. So these are vein cells. So if you infect them with Nipah virus, they look pretty happy and healthy. So to remind you, veins don't really express high levels of the Nipah receptor FNB2. If we look at arteries, something really crazy happens. So in just 24 hours, the arteries are destroyed. They form this weird spider web that's almost peeling back from the plastic of the plate. Like you can see the webs of the spider web. You might ask what's happening, and there are actually 23 individual cells are fusing together, and all their nuclei are actually bunching together like a bunch of grapes. I thought this is really quite weird. So you might think maybe this is a bit of a big claim. How can we prove this? So we turn to live imaging. So in this experiment, we have a co-culture of wild-type non-fluorescent artery cells and green GFP-expressing artery cells, such that when they're infected with Nipah virus, if they're really fusing, green would diffuse from one cell to the other. And this is actually technologically quite complicated because it's the second time in history that live imaging had been done in the BSL-4. You might ask why, because live imaging is so common in many of our labs. It's because if you guys have ever heard of the song Hotel California by the Eagles, you can always check in, but you can never check out. So when you bring in this microscope into the BSL-4, if something would happen to it, Zeiss or Nikon is not coming in to fix it. It's basically just to stay in the BSL-4 for the rest of its natural life. So it's basically this microscope can only be kept now in the level 4 because of the single movie, essentially. So in this movie, it's going to show two types of cells, GFP positive cells in white and wild type cells in black. When I press play, you can see new white spots are appearing. So that's because GFP is diffusing from one cell to the other. But look, the nuclei have a life of their own. And they're migrating to the center of these cells where they form these large clumps. So this is not one nucleus, it's many nuclei aggregated together. Now at this point, there's almost no black spaces on the plate because almost everything has fused into several massive webs. And then you can see that Nipah completely consumes these cells. And I think it's quite remarkable because it's just within 8 hours, which is a time maybe from now until dinner that all this happens. So for those of you who watched Contagion, I think this is what's happening in Kate Winslet's arteries. And if you haven't watched Contagion, unfortunately, she doesn't make it to the end of the movie, although she was one of my favorite characters. So we then asked, what is Nipah really doing to these arteries? So we take artery cells, we either infected with Nipah or as a control Sendai virus. So Sendai virus is in the same viral family as Nipah, the paramyxoviruses, but doesn't cause human disease. So we think this is a well-controlled experiment where we're infecting cells with two viruses from the same family with different effects. We just do RNA sequencing. So to tell you about this, I need to give a bit of background that when many viruses infect human cells, they trigger the production of a cytokine called interferon, which turns on many antiviral defenses that slow viral replication. So for instance, if endothelial cells are infected with Sendai virus, the harmless one, it's well known that they produce interferon and it slows viral replication. But what's really quite remarkable is that in that whole movie I showed you where Nipah is entering, replicating in, fusing and killing those artery cells, The cells are completely brain dead and don't produce interferon. So this is a heat map where you can see that different viruses are being added in different columns, and yellow indicates high gene expression. And what you can see is that when you add Sendai virus, which is the control, although Sendai occupies less than 1% of the transcriptome, there's a massive response. So interferon, the antivirus cytokine, turns on in yellow, and a whole suite of antiviral defense genes turns on. What's quite stunning is that although Nipah, when it infects these arteries, is causing all this destruction, it occupies 1 out of 6 mRNAs, which is staggering. Imagine 1/6 of your mRNA just becoming pure virus. Look at that. There's no interferon being produced or these antiviral response genes. So the analogy I like to use is that Nipah is like a stealth fighter, that when it's entering, replicating in, fusing, and killing these arteries, it's like a stealth fighter evading the artery's ability to detect it. You might think, wow, that might be interesting, but maybe we're really cherry picking these genes to show this result. So this is now looking at all genes in your transcriptome, a volcano plot, where each dot is a single gene. See, when you add Sendai virus, massive numbers of genes turn on and off. We add Nipah virus, 0 genes across the whole transcriptome are turning on or off. I think this is really quite profound. It's well known that many viruses inhibit innate immunity, but Nipah is absolutely stunning in its ability to completely kind of turn off the cell's ability to detect or react to it, even though it's already replicating at quite massive levels at this time point. So in summary, we can make over 90% pure artery or vein cells in 3 to 4 days of stem cell differentiation. And we use these artery and vein cells to find that these very exotic virus, Nipah virus, doesn't attack all blood vessels equally, but preferentially attacks arteries and causes them to fuse these massive multinucleated clumps that then rapidly die. And Nipah is extremely stealthy and doesn't change gene expression in this process. We have a lot of ongoing work, also focusing on vein development. So in this preprint, which is on bioRxiv, but I won't talk about today, we found that there's a key intermediate step in vein development where cells co-express an artery marker, SOC17, and a venous marker, apelin receptor. We found that the key signal that controls this is VEGF. So VEGF is vascular endothelial growth factor, known to many of you as a mitogen for blood vessels. And VEGF is critical for mesenchymal cells become these intermediate endothelial cells. But surprisingly, it's the inhibition of VEGF that creates veins. And we find this very surprising because VEGF is ubiquitous in media to generate endothelial cells or to expand them. But we think it's VEGF inhibition that's really inducing a venous identity. So of course, there's many questions that remain. So beyond arteries and veins, there's a large assortment of different endothelial cells like lymphatics, capillaries, and so on, which you can't make in vitro. So that's of course an important goal for the field. We're very curious about the mechanisms through which Nipah evades an artery's innate immune system. I think it's actually quite profound that Nipah can do this because there's actually only 6 genes in the Nipah virus, which I think is quite remarkable that something like only 6 genes can kill almost 60% of people. And so it should be, in theory, if something has only 6 genes, fairly easy to figure out how it's blocking innate immunity. Of course, those are famous last words. We also think that it's very cool if we could use this technology to make gene therapy stealthier. Because as you know, one major side effect of lentiviral or AAV gene therapies is that they activate innate immunity. And many companies have invested many resources in trying to make stealthier gene therapy vectors. But I'm always thinking, well, if you think of viruses like Ebola or Nipah, they have had tens of millions or hundreds of millions of years to become really good at this, or sorry, I should say tens or hundreds of millennia to become really good at this. So we can figure out which viral proteins or peptides inhibit innate immunity. I think those would be really helpful for gene therapy. And beyond Nipah virus, do other BSL viruses infect blood vessels? What do they do to them? In fact, our lab is working on Ebola virus, a Marburg virus, Andes virus, which is the hantavirus as a causative agent of the cruise ship outbreak that just happened. So stay tuned. We're working on all these viruses and hope to report back on what we found. And in case you're bored, this is a great book called The Hot Zone, which got me to science in 2nd grade. This is the book that actually popularized the Ebola virus and to the general public. In fact, I think many BSL scientists are actually not that happy with this book because this is why everyone knows about Ebola, but say not Nipah or so on. But it's a great intro in my view. And I want to thank our many collaborators, including Joe Prescott for the work in the BSL-4, who's a great partner and colleague, Lei Tang Ang, who's contributed greatly to stem cell work, as well as Kevin, Elena, and Xiaochen, who are key trainees who carried this on. So yeah, I'm happy to pause here for some questions before moving on if there's time to a second part.
Hi, Kyle. This is Lee.
Oh,
Can
hey
you hear
Lee.
me?
Yeah,
Hi.
thanks so much for taking the time to attend today.
It's fantastic talk. I wonder, is there any differences in terms of the virus and its infection effects on human cells versus mouse cells? Have people done things before in mouse models or other models that may behave very
Yes,
differently?
that's a truly outstanding question. And actually, that's why our lab exists in some part. All these BSL-4 viruses basically only cause human disease.
Mm.
They or in non-human primates, which are the gold standard model, if you give Ebola or a Nipah to a mouse, it does nothing, actually, which is really amazing. Actually, only under very, very rare conditions, like if the mouse is very young or it's immunocompromised or extremely old, will Nipah have an effect. But these are human pathogens, and that's why we think that, unlike many things in biology where you can model it in a mouse, won't, it doesn't really do anything, which is why these
The,
human cell
the,
models are so important.
well, absolutely. That even makes your work much more important because now we have a model to study them, and that's fantastic.
Yeah, thanks for saying that.
Hey, Kyle, very nice
Hi,
talk.
hi,
This
Jeremy. I,
is,
I actually I don't know if I've met you before, but actually I read your paper as an undergrad with Sung Kim when I was a summer student, actually. So it's kind of interesting attaching your face to this name that I followed for like over 15 years now,
that's
actually.
very kind. I just have more
Yeah,
gray hairs.
yeah,
Um, so one thing I'm wondering, it's super interesting that you can kind of separate into an artery and a vein fate. But I'm
yeah,
wondering if you have an opportunity to understand a really challenging pathology that we deal
yeah.
with in
Okay.
my world of neurointervention, which is arteriovenous malformations. So there's clearly some hiccup, and I'm wondering, what
Yeah,
if you induce a venous fate in something that's already fated towards an artery? Do you start to get a nidus? I'm just curious if you thought about that.
I would love to talk with you, in fact, because I actually got an MCHRI grant on AVMs and never did a single thing with it, and the grant is over. But I find AVMs really fascinating because I know that clinically it's described as a nidus, but from a
Mhm.
dev bio perspective, like what you were take before, I'd be like, I don't know what that means, like molecularly. Like in fact, with a
Mhm.
science paper that did like single cell RNA seq of an AVM, and they just called all the cells in the middle nidus. And I was like, oh, I was really hoping like if you could look whether they have artery or venous markers, instead of just like, calling it the third class, or maybe it is like a third class. Um, and there's also a lot of interesting work in mouse models where people deleted the AVM risk genes like SMAD4 in
Mhm.
either arteries or veins. And basically, there's a paper in Circulation Research I can send to you that only if you delete SMAD4 in a vein in a mouse does it form an AVM-like structure, but if you delete SMAD4 in an artery, it doesn't. So it's clear, it seems to suggest that the vein is the origin of the AVM, but what is
Interesting.
happening to it to make it that it's not clear. I mean, there's a whole subfield of AVMs in the vascular biology. And of course, like the culprit is, quote, cell cycle, as you can imagine. So they're like, oh, if you give a CDK inhibitor in like a mouse model, it looks better. It's like, well, I don't know if that's the most specific, but I would be very happy to find ways to interact with you and your colleagues. Like this is like the perfect um, reductionist human system, I think, to tackle these questions.
Very interesting. I'll ping you separately.
Yeah, I would love to. I would love to chat. Yeah, and I think there's a question from Jessica. Yeah, thank you so much, Jessica, for your thoughtful question. We haven't worked on the cell-cell fusion mechanism, but it has been studied. So it's actually quite cool. So let's say that I'm an artery cell infected by Nipah, which I hope never happens. The Nipah envelope protein would be displayed on my cell surface because when a viral genome will come up under the plasma membrane, it will pinch off and bud off. That's what's supposed to normally happen. But if that doesn't happen, I'm just an artery cell with Nipah envelope proteins on my surface. Now I'll come on next to someone I like Mateo, maybe, if he's an artery cell. And basically, the Nipah envelope protein on my surface will just recognize his F1B2 and mediate fusion. So the viral envelope proteins evolved to mediate like viral particle to cell membrane fusion, but it can also just mediate cell membrane to cell membrane fusion. And the other viruses are known to make syncytia, not just Nipah, but Nipah is the only one that clinically is known to cause endothelial syncytia, to my knowledge, in pathology reports. So it's kind of weird. You might ask evolutionarily, why was there some selection for this to happen? Of course, we don't know if it's purposeful, but one speculation we've had is a normal virus. Let's say I'm an infected cell and I have to bud up a particle and infect Mateo. Sorry, Mateo, I'm not directing all this aggression at you, but just you're a convenient character for me today. In the bloodstream, before the virus particle reaches Mateo, an antibody could intercept and interdict it and neutralize it. But if I'm just sitting next to Mateo in a blood vessel and I just fuse with him directly, there's kind of no time for an antibody to kind of neutralize that. So I think that's very interesting to think about in vivo, direct cell-to-cell transmission of a virus, as opposed to typical cell virus budding and then cell entry. Yeah, if that's that, I'll move on to a very short vignette about the brain. That the brain we often think of as a single organ that comes from a common progenitor called the null ectoderm. But we found there's not one, but actually two separate brain progenitors. One makes a forebrain midbrain, and the other makes a hindbrain. And this new view of the brain as not one organ, but actually a composite organ of two different sources, we think has broad implications for development, differentiation, and evolution. So just to orient you now in this map, we're looking at the third branch, the ectoderm in purple. Before starting, I want to thank my terrific lab members, Carolyn and Ryan. Of course, Mateo has been very helpful to assist Ryan in some of these experiments I won't talk about today, together with various trainees in the lab, Rachel Kang, Hadia, and Rachel Solomon. So to take a step back, the brain has three main parts. The forebrain midbrain do fancy stuff like thought, reasoning, and memory. But the hindbrain, which makes up most of the brainstem, this controls the life-sustaining functions like breathing, heartbeat, wakefulness, movement, and sleep. I'm going to focus on the hindbrain for today. You might ask, why do we care about the hindbrain? It turns out there's many methods to turn stem cells to forebrain midbrain, but it's been a lot harder to turn stem cells into hindbrain cells. Thought, that's a bit curious. Why would that be? And it also turns out that the hindbrain is the only part of your brain need to stay alive. So what do I mean by this? I'll tell you about Mark the Headless Chicken. So in 1945, a farmer beheaded his chicken. Of course, this is before there was AppLac that existed that prevented you from doing things like this. But because the farmer was not a scientist, he did not cut off the whole head cleanly, but rather cut off only the front part. And this only had a hindbrain. I guess my question for you is, do you think that this chicken survived with only a hindbrain? And if so, for how long? Okay, Mateo's smiling. I assume that, what do you think, Mateo?
I really don't know. Sorry, it was
He's smiling. 1.5 years. So
That's
it
a
was
lot.
quite a long time. So I
Wow.
tell people this is an incredible experiment of sorts because it really means that every single second you're staying alive, it's your hindbrain that's keeping you alive. So that's why we want to work on it. So the current model in developmental biology, first proposed by this guy called Peter Newcoop in 1952, is in development, you form a common progenitor called the null ectoderm, and this makes the forebrain, the midbrain, and the hindbrain. But classical studies have suggested that maybe not all null ectoderm cells are the same, and some might be biased already to make different brain regions. In fact, our data supports this historical view. We find that there's not one, but two separate brain progenitors. One that we call the anterior null ectoderm, anterior meaning front, which is the forebrain midbrain, and that the current model of brain development of inhibiting TGF-beta and BMP really describes how to make this first fork. We find that there's a separate posterior null ectoderm in green that makes the hindbrain. We think that this explains why it's been so hard to turn stem cells to the hindbrain, because we didn't know that the hindbrain came as its own separate developmental source. You never know to turn off to get there. The analogy I like to use is like you're navigating down a main road leading to the forebrain and midbrain. If you didn't know there's a side road leading to the hindbrain, you'd never know to turn off and to get there. So how do we discover that there are these two separate brain progenitors? Because this is deeply counterintuitive. Well, we simply just looked for it by staining. So this here is a photo of a mouse embryo. So early in development and gastrulation, you don't see a head or a tail at E7.5, 7.5 days. Here we're staining for two genes, OTX and GBX2. And I hope this image will convince you there's not one brain progenitor, but actually two separate brain progenitors. One expressing this gene OTX2 in red, the other called GBX2 in green. And this is building on classical studies of the expression of these genes in the 1990s. So I hope this image will really convince you there's not one, but two separate brain progenitors expressing different genes and located at different positions at this stage. So then we ask, do these two different brain progenitors give rise to different parts of the brain? So use a trick called genetic lineage tracing, we can permanently label the second brain progenitor, the posterior neural ectoderm, and ask, does it give rise to the entire brain, which would invalidate our model, but give rise only to the hindbrain? So the next slide, all the progeny of this posterior ectoderm will be labeled red. So this is a photo of the mouse brain. You can see there's the forebrain, the midbrain, and the hindbrain. And what was really remarkable is only the back half of the brain, the hindbrain, is in red, indicating it came from the second progenitor, the posterior neuroderm. So I think this image is very surprising because if you took away the colors, you would just see one gray brain. If you look back in developmental time, which is what lineage tracing lets you do, you see the back half of the brain come from a totally different source from the front half of the brain. So to summarize, the brain you can think of as like a lake with a unitary body of water. But even a lake, if you look upstream enough, actually can come from two different streams that join together. So after discovering that these two brain progenitors exist in vivo, we then developed methods to create them in vitro. We found that the current signals for neural induction, inhibition of TGF-beta and BMP, describe how to make the anterior neuroectoderm, leave an open question of how to make the posterior neuroectoderm in green. So in summary, if you take a pluripotent stem cell and dish and add the current signals to make brain cells inhibiting TGF-beta and BMP, you make these SOX2 positive anterior neuroectoderm cells. But they are not just neural cells. They express that first marker I told you about, OTX2, that anterior neural marker. Reciprocally, if you inhibit TGF-beta and BMP, but now add two additional signals, epinephrine and retinoic acid, you still make SOX2 positive neuroectoderm cells, but now they express the second marker I told you about, a GBX2 in purple. So in summary, although it takes many weeks or months to make neurons or brain organoids, we think in a Petri dish, the fundamental decision to become forebrain, midbrain versus hindbrain is made in the first 2 days. So having made these posterior neuroectoderm cells, we can then go on to make hindbrain progenitors. And to my knowledge, our terrific MD PhD student, Ryan Jokai, was actually the first to make these specific hindbrain progenitors of a region called ROMAmeters 5 and 6, expressing the marker proteins shown here. We then went on to take these hindbrain progenitors to make certain neurons. So there's many kinds of neurons in the hindbrain because it controls life-sustaining functions, but today I'll focus on hindbrain motor neurons. So these sit in your brainstem or hindbrain and project through the cranial nerves to control your face, jaw, eye, tongue, and neck muscles. So you may ask, who cares about these? These are critical for eating and swallowing, and also facial movements and speech. And very sadly, the degeneration of these hindbrain motor neurons in diseases like ALS or SMA, which estimate the pediatric disease, can lead to the inability to eat and choking, which can be fatal. So we've now succeeded in creating these hindbrain motor neurons. They have these beautiful neurites expressing MAPT or tau. They express the motor neuron transcription factor ILT1 in the nucleus. And they produce acetylcholine, which is the key neurotransmitter made by motor neurons. These hindbrain motor neurons were also electrophysiologically active and fired action potentials as shown here. I just want to add that we're not the only ones who seem to believe that there's two separate brain progenitors. And I want to encourage all of you that there's a beautiful bioarchive preprint from Agnete Kirkby's lab in Copenhagen, my apologies, in Sweden, where they also found that there seem to be two separate brain progenitors. The last thing I want to touch on are the evolutionary origins of these, because in the field of evolution, of course, the origin of the brain has always been a very compelling kind of ancient question. As the most complex organ, when did the brain come about? And we're very excited that we found these two brain progenitors existed not just in human and mouse, which are mammals, but they're found in chicken, zebrafish, and even acorn worms. We think the fact that they're found in acorn worms is really quite remarkable because acorn worms are deuterostomes that diverged from us over 500 million years ago. So this is work done with Chris Lowe as an Evo-Devo specialist in Stanford's Hopkins Marine Station. So this is a photo of an adult acorn worm. I hope you agree it looks nothing like a human. But if you look in development, it looks just like those mouse that showed you with this anterior OTX positive anterior neuroectoderm in the back, this GBX positive posterior neuroectoderm. So in summary, it's often thought that the common progenitor for the whole brain. We think there's two separate brain progenitors, one for the forebrain midbrain, the other for the hindbrain. We think this has very broad evolutionary implications. If you go further back from acorn worms to cnidarians, which are like jellyfish, it's actually long been known in a jellyfish that there's not one nervous system, but actually two physically separate nerve rings, one in the front, one in the back. And I think that the idea that the brain is one organ may be a historical anachronism because people studied mammals first, where obviously we have one brain. But we think if science take a different turn, if people had just studied jellyfish first, they'd say, well, clearly this animal has two separate nervous systems that maybe came from different progenitors. Then when it came to us, if God could reinvent things, God would make them a common progenitor, but evolution often just takes the existing progenitor field and spatially kind of push them together so that the brain could synapse and communicate more efficiently. Of course, this is speculation, wild speculation, and we were, of course, forced to remove this from our paper by reviewers, probably rightly so. And this knowledge has allowed us to differentiate human stem cells, the hindbrain progenitors, and hindbrain motor neurons. So, of course, there's many unanswered questions. So we find that by the end of gastrulation, there's two separate forebrain, midbrain versus hindbrain progenitors. But is there a pan-neural progenitor that exists? And we cannot exclude this possibility because in development, it's really hard to prove that something doesn't exist. And finally, although we find that the forebrain, midbrain versus hindbrain come from different progenitors, a logical question is, where does the spinal cord come from? And we find that there's actually a third separate progenitor for the spinal cord and actually a fourth separate progenitor. So it actually seems to be that the central nervous system, from your brain to the spinal cord, the adult appears as one contiguous tissue. But we think that in development, there's actually four individuated progenitors that are all converging to make different parts of it. So in closing, I'd like to thank my wonderful lab members who led this brain work, including Carolyn, Ryan, and Rachel, our terrific lab members, our collaborators for the hindbrain project, including Chris Lowe, Marianne Bonner, Michelle Mange, Dan Wagner, and Chris Garcia, and support from various sources. And I thank all of you for listening. Thanks so much. I'm happy to take questions if there's any.
Fantastic talk, Kyle. Thank you so much. Anyone who would like to ask a question, they can unmute and ask directly or write in the chat, or maybe I can read something that is in the chat. I don't know if you have already seen this. What do you think? Oh, you did. Okay, sorry. Maybe I missed that.
Yeah, it's fine. As often as there's no questions, I'm also happy to give people the rest of the few minutes back, or people can rest before their 1 o'clock too. Up to everyone. I'm always happy to take questions or happy to be contacted by email too.
Well, I think yes, if there are probably no questions, feel free to email directly Kyle. And thank you so much for the fantastic presentation, Kyle.
Thank you everyone for taking the time to attend. I know there's many seminars and things competing for people's time, and I'm very grateful. Thank you.
Bye, everyone.
Okay, thank you. Bye-bye. Thank you, Matteo. And Matteo, always happy to talk if you want to talk about the vasculature, because that came up in Gene's talk yesterday that he's missing the vasculature, right? So
absolutely. I'll come to we'll grab a coffee and chat.
Or I can come down. I'm happy to always talk.
Sounds great. See you soon, Kyle. Bye-bye.
Bye.